everyone, Professor Davis here, and so let's take a look at your lecture final. So we're going to review the 9 chapters that we went through for this Bio 1 course. So starting with chapter 1, we see that you need to understand the definition of homeostasis. Homeostasis means that you maintain a steady state internally. In other words, it helps you keep a balance inside. Homeostasis is one of the things that is required for living things. Now, some other things that are required for life, these are the characteristics of life, is that life is organized, it's able to reproduce, respond to stimuli, assimilate or use energy, evolve, do homeostasis, which we just talked about, and life forms ecosystems. So you need to make sure that you can identify the characteristics of life. It's probably going to be a question that says like, all of the following are characteristics of life except. Now, we also need to know what the different kingdoms and, well, what the different domains are, and then kingdoms for living things. So the 3 domains are bacteria, archaea, and eukarya. Eukarya also contains kingdoms as in protista, fungi, plantae, and animalia. Make sure you know examples of each. So bacteria is going to be the main ones that live. Bacteria lives pretty much everywhere. Things like E. coli, strep, stuff like that. Archaea are your real extreme bacteria. They like to live in extreme environments like your thermophils, acidophils, that kind of thing. In eukarya, when you look at the different kingdoms, animalia, of course, is the animal kingdom, so any animal. Plantae are plants. Fungi is going to end up being fungus, so like mushrooms, yeast, that sort of thing, molds. Protista is like the junk drawer of the kingdoms. Protista has some that are animal-like, which are your protozoas. We also have some that are plant-like, which are your algae. And then we have some that are fungus-like, like your water molds. But those are all in the group Protista. We also need to remember that there's an organization. When it comes to living things, we see that the basic unit of life is cells, and then cells can work together to make tissues. Tissues work together to make organs. Organs work together to make organ systems. Those organ systems make organisms. Organisms living together make a population. Populations then make a community. Now, when we get past the community side, this goes to the ecosystem, which contains the living and non-living things. So an ecosystem has the biotic, which are the living things, and the abiotic, like water, air, soil, that makes up the ecosystem. And then, of course, last but not least, the biosphere, which is the whole planet Earth. Also in chapter 1, we talked a little bit about the scientific method. When we look at the scientific method, we do see that sometimes when things get proven over and over again in a type of experiment, this creates a theory. It's a well-supported, generally accepted principle. This creates a theory. A hypothesis, remember, is a tentative explanation. And the scientific method, we're going to see that we need to know the different groups that we use for the scientific method, variables, steps, and how we interpret what you're given. And so when we look at this, guys, the idea here is that with the groups, when we are using the scientific method in a proper experiment, remember that you're going to have your experimental group. Then you have also your control group. So remember, review what experimental group and control groups are. Also your variables, your experimental variable is the one you change. The responding variable is the one you measure. When we look at the steps of the scientific method, remember that this deals with observation, hypothesis, experiment, and then conclusion. And then our conclusions, a lot of times we can use that to create a new hypothesis or adjust the one we have. But if it's proven over and over again, this is what leads to a theory. Make sure you understand what kind of questions we can answer with the scientific method. Remember, we can't answer every type of question, especially those that are subjective. Like we can't answer what the best chocolate chip cookie recipe is, because to me, it may be one thing, to you, it's a different. Like I don't like nuts in my chocolate chip cookies, but some people do. And so some of those types of questions cannot be answered. Now, chapter 2 is the chemistry chapter, and so there's a little bit more we want to take a look at here. Remember that when we reviewed chemistry, the subatomic particles that make up an atom are your protons. Protons have a positive charge. We also have neutrons, which are neutral. Remember, the protons and the neutrons are both located in the nucleus of the atom. The electrons are found outside of the nucleus and they have a negative charge. The electrons are what's going to determine what kind of bonds that atoms are going to make with each other. So ionic bonds are going to be where they complete a transfer of electrons. So like one becomes more negative because it gets an electron and the other one gives it away, so it becomes more positive. This creates ions, and then opposites attract. A positive is attracted to a negative. On the other hand, when we have polar covalent or even nonpolar covalent, covalent bonds are where they share. Sometimes they share equally and sometimes they do not. So polar covalent bonds are when they do not share equally. So it's an unequal sharing, and this is like water. Polar covalent bonds are going to have a kind of pole setup where one side's a little more negative and the other side's a little more positive, thus the polar part. Nonpolar covalent bonds have equal sharing, and so there's not a pool based on their positive or negatives, and that's something like methane. Another type of bond that takes place between molecules is called a hydrogen bond. That's what's unique about them, is they are between molecules. Water molecules will have this between them, as well as DNA molecules. We also looked at the properties of water. Remember that water likes to stick to things and itself, cohesion and adhesion. This is what helps create something called surface tension. We also see that water is a universal solvent, meaning a lot of stuff can dissolve in it. Ice is also less dense than liquid water, and this is why it floats. It takes a lot of energy in order to heat water up because it has a high heat capacity. And to get it to heat up enough to turn into a vapor, it has a high heat of vaporization. Now this is good because we don't want water to be able to change temperatures really quickly, because that would affect us since the majority of our body is water. Water has polar covalent bonds within the molecules, but it has hydrogen bonds between the molecules. This is a key statement because water does have polar covalent bonds between the hydrogens and the oxygens, but one water molecule will bond to another water molecule with hydrogen bonds. Okay, so make sure that you understand the difference there. Continuing in chapter 2, we talked a little bit about buffers. Buffers help minimize the fluctuations of pHs so that something doesn't become too acidic or too basic. Remember, the pH scale does run from 0 to 14. 7 is considered neutral. That's where pure water sits. Anything less than 7 is considered acidic, an acid. Anything more than 7 is considered a base or basic. It's also known as alkaline. Acids will donate positive hydrogen ions, which are going to be protons, and bases donate OH negative, which are hydroxide ions. Chemical reactions occur when bonds are broken apart, and so when these bonds that are made between atoms are broken, then we're going to see that it's a chemical reaction that takes place. Remember that some of these can end up being sped up if there's heat or slowed down if it's cold. When we talked about chemical reactions, there were some terms. We had anabolic chemical reactions. Anabolic means we were building something. Another name for that is called synthesis, where we build a molecule. Also, when you build something, you have to put energy into it, and this is why it's called an endergonic reaction, because energy went in. On the other hand, catabolic is where we break things down. When we break things down, that's also known as degradation. We're breaking it. And when we break bonds, energy gets released. And so this type of reaction is exergonic, meaning that the energy exits the compound. Okay, so anabolic, endergonic, and synthesis are all related, like interchangeable, whereas catabolic, exergonic, and degradation are also the same thing. Now, the very second part, like the big second part of chapter 2, dealt with the macromolecules, and we did a lab over this as well. This was lab 3. And they looked at the 4 types of macromolecules and their building blocks. Carbohydrates have the building blocks of monosaccharides. Proteins have the building blocks of amino acids. Lipids are built out of fatty acids and glycerol, and nucleic acids are built out of nucleotides. One thing to note is organic molecules like these, like carbs, proteins, lipids, and nucleic acids, are what we consider organic molecules, meaning they are going to have both carbon and hydrogen present. So carbon and hydrogen are present. If carbon is missing or hydrogen is missing, the molecule is an inorganic molecule. Some important polysaccharides that we have are cellulose, chitin or chitin, glycogen, and starch. This is probably a question that says all the following are polysaccharides except. Important lipids are things like steroids and phospholipids, which are found and make up the majority of your plasma membranes. Some important nucleic acids, of course, are DNA and RNA. Now, proteins, there's a lot of proteins that are super important, but the big thing with proteins is they all have different kinds of jobs, and that's what makes them important. Some proteins are there for structure. Some help with transport, like moving things. Some proteins act as receptors. They may act as a channel or carrier across the membrane. We may use some proteins for identification, and a lot of proteins are used for enzymes. And remember, enzymes speed up chemical reactions, and we'll talk a little bit more about them a little later. All right, moving on to chapter 3. And guys, I know these slides have a lot of information on them, but the whole point is that you can review this stuff. So the cell theory, guys, was developed by a scientist named Virchow. He said that all living things were composed of cells. So there are 3 parts to this theory. So all living things are composed of cells. Cells come from pre-existing cells, and a cell is the basic structural and functional unit of life. Okay? These things are all true. We know they're all true. The reason this is still a theory and not the cell law has to do with the fact of cells come from pre-existing cells. Because in science, we cannot actually pinpoint exactly where that first cell came from. Remember, with cells, we have 2 main types. There are prokaryotic cells and eukaryotic cells. Prokaryotic cells have no true nucleus. They only have an area called the nucleoid region. This is where their circular DNA is found. Remember, prokaryotic cells also do not have any membrane-bound organelles. They only have ribosomes. So these are a more simple type of cell, like bacteria. Eukaryotic cells, however, are more complex. They have a real or true nucleus. That's where the DNA is located. They also have membrane-bound organelles such as rough ER, smooth ER, chloroplasts, mitochondria, Golgi. They have all those different organelles present. There's also a difference here when we talk about eukaryotic cells. They can either be put into, a lot of times we say plant or animal cells. When we look at plant cells have a couple of structures that animal cells do not have. Plant cells have a cell wall. They also have chloroplasts, which are the organelle that does photosynthesis, and they have a large central vacuole. These things are not found in animal cells. But animal cells have 2 things that plant cells do not have. They have lysosomes, which are going to help digest and clean out, recycle things in the cell. And they also have centrioles, which are important during mitosis. So let's talk about a few of the organelles and what they do. The chloroplasts, guys, are important because they're the site of photosynthesis. They are where photosynthesis takes place. Remember that photosynthesis makes carbohydrates, sugars. The mitochondria is also really important because it's the site of cellular respiration. In other words, a lot of times we say that the mitochondria is the powerhouse of the cell because it helps make the energy in the form of ATP. We also have ribosomes. Ribosomes are important because they are the site of protein synthesis. This is where proteins get made in the cells. And then we also have the plasma membrane. Guys, the plasma membrane, if you'll recall, is selectively permeable. It doesn't allow anything and everything to move through. It picks and chooses. It's also the separation between the inside and the outside of the cell. Now, with this plasma membrane, remember that the plasma membrane has what we call the fluid mosaic model. This is because it is kind of a semi-fluid. It's like an ocean of fats. Okay, it's like an ocean of lipids, those phospholipids that have the head region with the two tails. The head is what we call hydrophilic, meaning it likes water, and the two tails are the polar fatty tails. They are hydrophobic. They do not like water. Also embedded throughout this lipid sea are proteins. This is what gives it the mosaic side, because there are proteins and cholesterol kind of spread out throughout the structure. Now, why would we have some cholesterol in there? Cholesterol is another type of lipid or fat, and it helps maintain the shape and rigidity of the plasma membrane. Now, when we talk about the membrane, remember the membrane does separate the inside of the cell from the outside of the cell, and it is going to help control what moves in and out, okay? With this, it does mean that there is movement of water that potentially does occur through the plasma membrane. This is known as osmosis. Osmosis is the movement of water from high to low, okay? So it's going to move from a high concentration to a low concentration. This movement of water is also based on what we call tonicity. Tonicity is the relative concentration of a solute, the stuff that's dissolved in the water. And so because of this, we get three types of solutions that could be present. Isotonic solutions mean that they have equal concentrations of solutes on both sides of the membrane. So in other words, the inside and outside of the cells are the same. Guys, iso means same. On the other hand, hypertonic means I have a higher concentration of solutes outside of the cell. So water is going to move out of the cell, and this causes the cell to shrink. If it's an animal cell, we call this shrinking crenation. Looks like a little sticker, like all these pointy edges. And in a plant cell, we call this plasmolysis because the plasma membrane shrinks or pulls away from the cell wall. Hypotonic solutions are when there's a higher concentration of solutes inside of the cell. And because of this, water moves into the cell and the cell begins to swell. The cell will eventually burst if we're talking about it being an animal cell, and we call that hemolysis. On the other hand, in plant cells, because they do have the cell wall, this just creates something that we call turgor pressure. Another thing about this plasma membrane is sometimes we need to transport large amounts of substances across the membrane. If we are taking these large particles into the cell, it is called endocytosis. So endo is going into the cell. And there are three types of endocytosis. There are penocytosis, where they're going to take in a lot of small particles at once, and this is known as cell drinking. There's phagocytosis, where it eats or takes in large particles. And then there's receptor-mediated endocytosis, which means there's receptors, and once the receptors have been activated, they bring them into the cell. On the other hand, when we want to move large particles out of the cell, we want to send them out. This is called exocytosis. So exocytosis is where we send the molecules out of the cell. All right, so now we're going to talk about chapter 4. Chapter 4 is where we started looking more at energy and how energy is going to be important to the cells. So guys, all energy on planet Earth comes from the sun. Okay, so all energy ultimately here on Earth originates at the sun. It does move through the ecosystem through the process of producers. So plants are able to collect the information directly from the sun, creating food. Then consumers can eat that, whether we eat plants or we eat animals who eat plants. And then eventually these things die and decomposers recycle different parts, but the energy is not one of the things that gets recycled, just the nutrients. This brings us also to then enzymes. Enzymes have active sites where substrates bind and help with these chemical reactions. Enzymes are important because they speed up chemical reactions. They make them go faster. But enzymes can be affected by a number of things. Things like pH, that whole idea of acids and bases. Also enzyme and substrate concentration, depending on how much enzyme or substrate we have, could affect how it acts. And then, of course, also temperature. Temperature can also play a big role with enzymes because enzymes, if they get too hot, they denature and they unwind. They lose their shape. Now, the main type of energy that our cells use, the currency, like the money that they use for energy, is called ATP, which stands for adenosine triphosphate. This means there's an adenosine with 3 phosphates attached. That's where the tri comes from. This is created or converted energy that is done by our cells in the mitochondria through what we call cellular respiration. Now, we can do cellular respiration without oxygen, and that's known as anaerobic cellular respiration. The problem with anaerobic cellular respiration is it does not create much ATP. It only makes 2 ATP at a time, and it gives you a short burst of energy, but it's not very long lasting. It also produces some toxic byproducts like lactic acid in our cells. On the other hand, if oxygen is present, we call this aerobic cellular respiration. This means that we can actually use the mitochondria and oxygen, and this creates a lot more ATP. We're going to look at the process of aerobic respiration in a little more detail. So cellular respiration generates ATP. This is for each glucose molecule if oxygen is present. The first step is glycolysis. It breaks down the glucose into what we call 2 pyruvate molecules, but it does generate 2 ATP. Those pyruvate molecules then go into the prep stage or the preparatory stage, and they're broken down into acetyl molecules that combine with a coenzyme A. There is no ATP that is made in the prep stage, but the prep stage does occur inside the mitochondria, so oxygen has to be present here. These new molecules with the acetyl coenzyme A enter into the Krebs cycle. This is going to turn twice since we have 2 of these molecules. And this is going to create some NADH, some FADH2, and they're going to carry the electrons to the electron transport chain. But as the Krebs cycle does rotate, since it rotates twice, we do see 2 ATP being made. So now we're up to 4 ATP. Now, all these electron carriers, NADH and FADH2, that have been carrying electrons from the 3 previous steps, from glycolysis, the prep stage, and the Krebs cycle, they're going to drop these off at the electron transport chain, and at this particular phase, it's going to generate anywhere between 32 and 34 ATP molecules. Now, that's a lot. This means that if you add the 2 previous to the low end on 32, you're going to see that we can make 36 ATP from each glucose molecule. That's a lot better than making just 2 that we saw with anaerobic respiration. Okay, so if oxygen's present, we can make a lot more ATP than if it's not present and only 2 are made. All right, to continue on with how energy is transferred, we're going to look at the different side of it in chapter 5, where we look at photosynthesis. Now guys, photosynthetic organisms are going to be things like plants, cyanobacteria, and algae. Now plants obviously are in the group Plantae, cyanobacteria are bacteria, and algae are part of Protista. But guys, they are going to do photosynthesis, and it's important for you to remember the actual equation for photosynthesis. Guarantee you there'll be a question about this equation. Remember that photosynthesis is where the plant is going to use carbon dioxide, CO2, plus water, H2O. They're going to have sunlight added in, and this is going to help them convert that carbon dioxide and water into sugar. That sugar is C6H12O6 glucose. Plus, it's going to release oxygen, O2. Okay, so our equation is CO2 plus H2O will then, with the arrow, be converted to C6H12O6 plus oxygen, which is O2. Now, this process of photosynthesis is going to take place in the chloroplast when we talk about it in plant cells or algae cells. This process has 2 steps. It has the light reaction and what we also call then the dark reaction. The light reaction takes place in the little poker chip structures inside the chloroplast called thylakoids. This is going to be where we take the water and we split it. So water has to go in, and with high energy, it gets split. This is where oxygen gets released, but the electrons are going to be transported along with the hydrogens to the dark reaction. Once they reach the dark reaction using NADPH as their carrier, we're going to then use the carbon dioxide, the CO2, and we will do 3 steps. The dark reaction is going to take place in the stroma, or the liquid portion of the chloroplast. We'll see the first thing is we have to fix the carbon because carbon is a gas, so we got to hold on to it. We then reduce the carbon into a carbohydrate, and then we regenerate the RUBP, which allows us to then fix carbon again. This is known as the Calvin cycle. So oxygen is given off as a byproduct, as well as creation of a carbohydrate or sugar. All right, that's enough about photosynthesis. Let's move on to chapter 6, which is mitosis. Guys, with mitosis, remember the purpose of mitosis is for growth and repair. This is a type of asexual reproduction. We have one cell that splits into 2 identical daughter cells, and it does it through a series of steps. We have prophase, metaphase, anaphase, and telophase. So remember, in prophase, the chromosomes condense so that you can see them. The nuclear membrane starts to split and disappear, and so does the nucleolus. The cell is getting ready to divide. In metaphase, the chromosomes line up in the middle at what we call the metaphase plate, or the equator of the cell. This is also when the spindle fibers attach to the chromosomes. During anaphase, the sister chromatids then pull apart to opposite sides. Okay, so they're going to pull apart. They actually look like little V's as they're being pulled apart to opposite poles of the cell. The cell also starts to elongate, and in telophase, a cleavage furrow in animal cells is produced, or a cell plate in plant cells forms because they have the cell wall. But this is known as cytokinesis. We also see that then the chromosomes start to unwind. The nucleus starts to reappear, and we are then going to have 2 new separate cells. However, they're going to look exactly the same, and they're going to look just like the parent cell. But guys, things can end up messing this process up, and those are known as carcinogens. They're cancer-causing agents. This is where cancer cells will grow uncontrollably. They will do this process over and over again without the normal checkpoints that normal cells have, and this is what creates a tumor. So carcinogens are what causes or triggers the cancer. All right, chapter 7. Chapter 7 focuses in on meiosis. We saw that mitosis is for growth and repair, but meiosis is for reproduction. It's a type of sexual reproduction. So when we look at this, remember mitosis has 1 division. Meiosis has 2 divisions. Mitosis has 2 identical daughter cells, whereas meiosis creates 4 unique cells. These cells are known as gametes, and gametes are egg and sperm. We see in mitosis, they are diploid cells, meaning they have a complete set, meaning 2 chromosomes for each. Whereas in meiosis, they create haploid gametes, meaning that the egg and sperm have half the chromosomes. Now, when you put the 2 next to each other, meiosis 2, not meiosis 1, but meiosis 2, looks like mitosis. The stages look very, very similar. But remember that meiosis is going to have a division where it's meiosis 1 followed by meiosis 2. And one of the main differences is during meiosis 1, homologous pairs of chromosomes form together, and they form what we call the tetrads. Tetrads can be seen in prophase 1, metaphase 1, and anaphase 1. In telophase 1, they have already pulled apart. We then see that the pairs do line up of these tetrads in the middle of the cell, and they pull apart. Then both of these new cells are going to go through the process 1 more time. They then go through prophase 2, metaphase 2, anaphase 2, and then telophase 2. At the end of telophase 2, we now have 4 cells, and each of those 4 cells is unique. And we've made egg or sperm. All right, when we get into chapter 8 is going to be where we start to look at genetics, but there's a little bit of kind of overlap between chapter 8 and chapter 7, because if meiosis doesn't happen correctly and eggs and sperm aren't made correctly, we sometimes have some abnormalities or some mistakes that get made in our genetics. And so when we look at this, karyotypes are used to tell us if there's any problems, what we call aneuploidy, meaning that there's not the right number of chromosomes. And if we have a monosomy, that's where we're missing a chromosome. And one of the best examples, and really the only example that survives, is Turner syndrome, where we have one X and we are missing the other one. So instead of having 46 chromosomes, this individual has 45. On the other hand, we can have trisomies where we have 3 copies of chromosomes instead of just the 2 copies we're supposed to. One of the best examples here is Down syndrome. They have 3 copies, chromosome 21. We also see Klinefelters could also be part of this, where they have an XXY, where they've got 3 copies. Also, when we look at our genetics based on our chromosomes, if you have an X and a Y, you are male. If you have an XX, you are female. So based on those sex chromosomes, an X and a Y makes them male, XX is female. All right, jumping into chapter 8, where we're looking at the genetics. Gregor Mendel, if you'll remember, is the father of genetics. He is the Austrian monk who did a lot of the testing and gave us these first couple of laws of genetics. The first law is the law of segregation, meaning that these chromosomes will segregate or split apart. So when we create egg and sperm, there's going to be a separation of the chromosomes where the mom's set goes one way, dad's is going to go a different way. There's segregation. However, the second law says there's independent assortment, meaning chromosome 1 doesn't care what chromosome 5 is doing. Chromosome 5 doesn't care what chromosome 12 is doing. This gives us a unique shakeup of the DNA where some of it comes from mom and some of it comes from dad in each of the newly formed egg or sperm cells. We then see with genetics that we have 2 main terms here where we have the phenotype and genotype. The phenotype is the physical appearance of the organism, and the genotype is its genes or genetics. So a genotype might be something like big T, little b, little b, big R, little r. Those are going to then be described as their physical appearance, where we might say that they are tall with blue eyes and curly hair. Okay, so that's the phenotype. We also see when we talk about genetics, there's dominant and there's recessive. Dominant traits are the ones that you see, and they are represented by a capital letter. So they can be represented where you have 2 capital letters, or you could have a big capital letter with a little letter. The little letter are the recessive traits. Recessive traits can be hidden by a dominant trait. So you have big T, little t. The little t is hidden. And these get passed on from parents. And if 2 little letters get put together, then you will actually see the recessive trait. So if the parents aren't showing that they have that trait, but they're carriers of it, their kids may have that trait and they didn't even know they carried it. So this might be where you have 2 brown-eyed parents have a blue-eyed baby because they were carrier of the blue-eyed trait. Okay, or 2 brown-haired parents have a blonde baby because they were carrying the blonde trait, the recessive trait. Okay, so this is what's really unique or cool about genetics. All right, this brings us to our last chapter, which is chapter 9. Now, with chapter 9, you need to make sure that you know the building blocks of nucleic acids as well as the types of nucleic acids. The building blocks of nucleic acids are nucleotides, and we already discussed that back in chapter 3. So the 2 main types of nucleic acids are DNA and RNA. So let's compare DNA and RNA. So DNA, the nucleotide, contains a sugar, a phosphate group, and nitrogenous base. The sugar that DNA has is called deoxyribose sugar. DNA is important because it contains genes that control your cell's functions. DNA is found in the nucleus, and remember, it is a double-stranded helix structure, kind of like a spiral staircase. RNA, on the other hand, also has a nucleotide as its building block, and it contains a sugar, phosphate group, and nitrogenous base. But the sugar is different here. It is a ribose sugar. We also see that RNA can come in three forms. We can have mRNA, which is messenger RNA, tRNA, which is transfer RNA, or rRNA, which is the ribosome RNA. RNA comes as a single strand, and RNA can be found in the nucleus and the cytoplasm. So this is what makes it different than DNA. Now, when we look at the nucleotides that make up DNA and RNA, there are things called purines, and that's the adenine and guanine. These are represented by the letters A and G. The pyridamines are going to be cytosine, thymine, and uracil. And guys, there's three because there's a difference here based on DNA and RNA. Cytosine is represented by a C, thymine is represented by a T, and uracil is represented by a U. So when we look at DNA, guys, DNA can contain A, T, G, or C, whereas RNA contains A, U, G, and C. So T is found in DNA, U is found in RNA. All right, so this brings us to some base pairing rules. When we're looking at how DNA pairs with DNA, DNA to RNA, and RNA to RNA, this is how it looks. So in DNA, A always pairs with T. It always does. G always pairs with C. Now this is when it gets a little trickier. When we have DNA to RNA, G pairs with C and C pairs with G just like normal. We also see that if DNA has a T, RNA will have an A just like normal. The difference here is that if DNA has an A, RNA has to have the U because RNA does not contain thymine. It does not have the T. Now, when we look at RNA to RNA, A always pairs with U and G always pairs with C. So I have a little example here. If I have T, T, A, G, C, C, A, is this DNA or RNA? Well, if there's a T present, this is DNA. So you could use this and you could practice your base pairing rules. If I'm going DNA to DNA, the two T's would pair with A would pair with T, G would pair with C, and the C's would pair with G. But if I'm using this DNA to make RNA, there would be some differences. So you can use this to practice. We also see that there is transcription and translation. Transcription is when we go from DNA to RNA. Remember, we copy down the code from the DNA to the RNA so the RNA can leave the nucleus. This RNA is then going to go and talk to the ribosomes, and it will be used to create proteins. That process is known as translation. So the last thing on here is translation. Translation is when we go from RNA to a protein. This happens in the cytoplasm at the ribosomes. And so this is going to be where we take it from the nucleic acid language to the protein language, where we go from RNA to a protein. So transcription happens in the nucleus, translation happens in the cytoplasm. All right, if you have any questions or concerns, please let me know, and good luck on your final exam.